into synapse in the chain ganglia. So if the tissue is innervated, if the tissue that is innervated is above the diaphragm, then it is almost always going to involve a synapse in the chain ganglia. If the tissue resides below the diaphragm, then the synapse is going to be in the prevertebral ganglia. Again, with a couple of exceptions. And these two slides are the four, well, there's actually a few more than that. These are the four general pathways that I think would get you through 98% of all autonomic anatomy. So the pathway on the left is for a tissue above the diaphragm. We have a synapse in the chain ganglion, and then, as you can see here, the postganglionic then goes back through the gray ramus into the peripheral nerve to go wherever it's going. On this slide, here we have a preganglionic efferent going out the ventral horn into the ventral root, spinal nerve, gray ramus. But this time there's no synapse. The preganglionic goes through the chain ganglion all the way down to a prevertebral ganglion, at which point it synapses and then extends to the target tissue. So this would be for the visceral tissues. This would be the tissues again below the diaphragm. So those are the only two hard ones. Those are the only two that makes the pathway can get a little complicated. The two general exceptions are the heart and the adrenal gland. The heart, you can see, has its own little cardiopulmonary splanchnic. Notice it does not re-enter the spinal nerve. Rather, it creates its own little pathway, which is, again, not typical. And then the adrenal medulla is actually considered neural tissue. It's actually considered autonomic tissue. Consequently, the neuron that you see here going all the way through, you can see it's also going down the splanchnics here. So notice this, that preganglionic goes all the way through both the chain ganglia and the prevertebral ganglia. And then it just sort of stops in the adrenal medulla, the assumption being the adrenal medullary tissue is the postganglionic neuron, because again, it's derived from neural tissue. And that's pretty much it. That's pretty much, and so now when you look at those larger diagrams, you should see a clear pattern there. You can pick any one of these, for example. You can see here we have a preganglionic, it's coming out. This is probably not the best one to pick, so a couple of these. There's a white ramus coming in. And then it goes up the chain ganglia all the way to the SCG, the superior cervical ganglia, where it then could innervate, for example, this would be a carotid plexus the eye. But what you'll notice is that synapse occurs in the superior cervical ganglia. It's still part of the chain ganglia. You look at this one, goes through the white ramus. Then there's a synapse in the chain ganglion. The postganglionic then returns to the spinal nerve to go off and innervate. Goes into the gray ramus to return to the spinal nerve, and then goes off to, for example, innervate pseudomotor or vasomotor structure. In general, so all of this should make sense to you now, but these aren't intended to be necessarily anatomically illuminating, but they do show the general structure of the sympathetic nervous system. So when it says T1 fibers pass up the sympathetic chain to the head, the idea here is that if all these fibers that come through here are going to go up through the chain ganglia, and you can see, for example, the eye, some other cephalic structure might be innervated. T2 goes under the neck, And then at T3, these structures are mostly thoracic, T3 down to T6. And then by the time we get to T7, these are abdominal pelvic viscera. This is just an illustration, a reminder of the cervical ganglia. The cervical ganglia, there are three of them. They have highly diverse and sometimes amorphous appearance. There's some strange connections. This is the ansa subclavia. Ansa just means circle or loop. We'll encounter that term a couple of times before we finish. But this is just an odd loop going around the subclavian that interconnects the middle and inferior cervical ganglia. So I don't think there's anything here. In fact, I think this slide was from the anatomy section. You know what we mean by the ANS being structurally divergent. They're more preganglionic. I'm sorry, they're more postganglionic than preganglionics. You know the general rules regarding the white and the gray raiment. You know the definitions of these terms here: vasomotor, pilomotor, pseudomotor. These would all be important. Sympathetic functions. We've made reference to the term splanchnic, so there are two major categories of splanchnic that we've talked about. There's that cardiopulmonary splanchnic, which is sort of a specialized one-off. Then you have the thoracic splanchnics, which are these in here. And then there are some other rather specialized splanchnics, and we'll talk about those. I think probably we can reserve some of this discussion. What tends to be the case, not to, as I say, not intending to complicate things now. There are sympathetic fibers in the pelvic region. We just don't call them pelvic splanchnic. But you should be aware that the idea that the pelvic splanchnic nerves and the parasympathetic, that does not come at the exclusion of sympathetic fibers in that region.
So the pictures are different.
How do you mean?
This one is different from the one
What's
that's
different?
before, like the ganglion.
Oh, they have a fourth.
There's a fourth one and a third one. Like there's four here, but on the slide, a couple slides previously, there's only three.
Yeah, we'll go with three. What happens is what we're going to find, and this will become clear in the anatomy. Well, actually, it's going to start, this anatomical process starts, and I don't think you see it in this illustration. A lot of, for example, these structures that we're calling ganglia here are just profuse networks that are wrapped around the aorta. And for example, we're going to find this happens in the carotid. There's a. Have you ever looked at the shielded cable, the cable that brings your all of your wonderful Comcast entertainment into your house? It's got a central wire, and then there's an insulator, and then there's a braided shield. Or you've ever picked apart a cable. What I'm suggesting is that these yellow things here are really just like braided shields. They're wrapped around in varying densities. And we could enumerate a number of these. We've talked about the celiac, the superior and inferior mesenteric. This is normally considered part of this. It's calling the aortic-renal. This is right there by the diaphragm, and this is right there by the renal arteries. But in reality, it's just a network of cords or wires wrapped around the blood vessel. An example of that would be here when we talk about the periarterial plexuses. The periarterial plexuses, once again, are these shield-like systems of fibers. Well, you can see some of it here. So this would be a plexus. And these dense networks are wrapped around vascular structures, sometimes other structures. And you can see some of that happening up here. Look at this, for example. Count the ganglia there. Anybody want to give that a try? How many ganglia do you see? Either 1, 2, 3, 4, 5, 6. But in reality, we name them 1, 2, 3. Right, and that's because they get so complicated and profuse. In fact, by the time we get to the pelvis, we just give up. The sympathetic and parasympathetic all jumble together, and it's impossible. So we just give them names and don't even attempt to disarticulate them. Here's that pathway to the adrenal gland. I just want to remind you that specialized flank to the adrenal gland is responsible for the generalized sympathetic response. The fact that when the sympathetic nervous system is excited, there's going to be release of adrenaline into the general circulation, and thus sympathetic activation tends to be much more systemic, much more broad spectrum than does parasympathetic. Yeah.
Is that true for like all, sympathetic activation? Like every time there's a sympathetic response, the adrenal glands are stimulated, or is that just in certain pathways?
No, I think, trying to think of what you mean by pathway specificity. There just isn't a lot of that in the sympathetic. Sympathetic can vary in respect to magnitude and amplitude, but it's fairly broad spectrum regardless. Remember, you even have a little mini adrenal gland in your brain that also releases adrenaline sort of non-specifically. That's one of the major differences between the two divisions of the ANS. Now, maybe what you're thinking of is receptor specificity.
Yes.
All right. So remember our general rules. I think we've already talked about this, but this would be true for the endocrine system and the nervous system of any type. The physiological response is determined by the receptor, not the neurotransmitter. So we could just release a profound amount of neurotransmitter, but if there are not receptors in the proper location for that neurotransmitter or for that hormone, have absolutely no effect. And we'll come back to that. We'll talk about the receptors in just a moment. So as you look at these pathways, you should be able to tell me what's going on here. For example, the pathway on the left, how would you describe this one? Except when I circled it, I circled both pathways. What pathway is that? What's going on there? Well, it's sympathetic, but which of those 4 pathways is it? Yeah, that's the one. When there's only one neuron. It doesn't feed into the basic rule. Our fundamental principle is all autonomic pathways consist of 2 neurons. Is that one? No. So that's certainly say that example. And then how would you describe, in terms of our organizational system, the way we classify those 4 pathways? What about the one on the right? How would you classify that? What type of tissue, or where is the tissue that's being innervated in this case? Exactly right. It's below the diaphragm. Of course, you see the viscera here, but what's the tip-off structurally? Synapse at the prevertebral ganglion. Don't worry too much about the discussion down here. A chromatin cell, that's another one of these general anatomical physiological terms that we're going to see all over the place. It just means that these cells like histological stain, and sometimes chromatin cells are special, and other times they're not. But these are chromatin cells in here in the adrenal medulla. All right, so much for any questions about the sympathetic nervous system. That's the hard part. There's really nothing. We've already talked about the general function of the parasympathetic nervous system. We've talked about parasympathetic tonus, the fact that under normal circumstances, you should be, as I'm sure you are right now in here, calm. In fact, I see some of you bordering on the comatose. You should be calm and sanguine and contemplating the higher law of the cosmos. So know the cranial nerves that have, because now we're talking about the craniosacral system, right? and this is what we've been talking about in the anatomy, know which of these cranial nerves carry parasympathetics in a piggyback fashion. We talked about the importance of the vagus. 75% of all parasympathetic outflow to the abdominal pelvic cavity is extended down the vagus. And so we'll have a lot more to say about that, but this is the vagus here, and you can see it going to the heart, the lungs, all the viscera here. And then this is, of course, the sacral portion of the pelvic splanchnic. The vagus in particular, I don't think it's indicated in this illustration, but because of the vagus has such a heavy workload, most of the time when you see reference to visceral afferents, that term comes up in the context of the vagus. And you can look at some of the complexity here. That vagus in some cases is going to wrap around vessels, wrap around other structures. It initially descends bilaterally in the neck, and then it comes together, and then it sort of weaves apart again. Very, very complex anatomical organization. But bear in mind that what that vagus is doing is it's sending signals to the heart, to the various portions of the lungs, and to the viscera, regulating blood flow, for example, regulating secretory activity. But it's doing that, once again, it's doing that in response to signals that it is receiving. For example, blood pressure signals, blood gas levels, and so on. So once again, the vagus is both efferent and afferent. Now, we'll talk more about it. I think we actually we've talked about these rules. I sometimes forget the order in which I put the slides. But remember, one characteristic feature of the parasympathetic nervous system is the preganglionics are very long. The postganglionics are sometimes just impossibly short. And in fact, the postganglionic neurons are generally located at the target tissue or the destination. It says here no pathways and neurotransmitters. So you know the pathways at this point. We need to talk about the neurotransmitters. Well, let's do that now.
So for the heart vagus nerve, when it says predominant parasympathetic, does that mean the vagus predominantly carries parasympathetic and not sympathetic? Or
Yeah, the vagus is entirely parasympathetic.
entirely?
But it doesn't, the implication when it said 75% to the abdominal pelvic cavity, the remainder is brought to that cavity by the pelvic splanchnic. So you can get it from the top all the way, the vagus handles all of that, and then at the bottom, that's a very precise anatomical designation. At the bottom, we have the pelvic splanchnic. All right, so this little illustration should look familiar to you. I just want to remind you, I think we had a quiz question about varicosities, right? So what we're looking at here ties a whole lot of things together here. Bring back physiology and anatomy, all sorts of things. This whole thing, this purple thing here, is an autonomic fiber. By fiber in this context, we mean an axon for the most part. So there's the axon, and you can see in this long, bizarre axon, there's no real neuromuscular junction, there's no neuron-neuron synapse. What we have is this long thread, and then at various points, the thread enlarges to become a varicosity. That's what a varicosity means in this context, sometimes called the tumescence. Now, here's the action potential coming down the autonomic membrane, and this should look familiar to you. What is this? Yeah, the voltage-gated calcium channel. We're talking about release of amines here, at least in this particular case, because we're talking about it doesn't really matter. We can say this is sympathetic. And so we have calcium-dependent exocytosis of, again, because we know it's sympathetic, it's norepinephrine. Norepinephrine into this region between the varicosity, and let's assume this is a smooth muscle or something like that. Now this should look familiar to you. What is indicated here? Yeah, we have a G protein receptor and the G protein. We talked a little bit about the components of that signaling. Any number of things can happen, but they're all going to involve a sequence or series of reactions, chemical reactions in the postsynaptic cell. Yeah.
This is a sympathetic.
Yeah, sympathetic because you can see the norepinephrine. Yeah. And we'll talk about the neurotransmitters in more detail. So here's another little slide involving some general rules regarding these neurotransmitters. So let's make sure we grasp this before we get into any further detail. All right, so we know that just about all autonomic pathways involve two neurons, preganglionic and postganglionic. Post. The preganglionic neurons in both pathways are cholinergic. Simple. Both parasympathetic and sympathetic, preganglionic is always going to be cholinergic. So this is a cholinergic neuron, and how we designate these neurons, we describe the neuron in terms of the neurotransmitter that is released. All right. In both pathways, the acetylcholine binds to nicotinic receptors at the ganglion. So far, so good. Very simple. Both pathways use the same mechanism for neurotransmission. In fact, the parasympathetic pathway is kind of boring because the postganglionic is also cholinergic. The only difference between the two is that the target cell receptor in the parasympathetic pathway utilizes muscarinic receptors as opposed to nicotinic. And we saw that in the previous illustration. So it's cholinergic all the way through in the parasympathetic. We have nicotinic receptors in the ganglia, muscarinic receptors in the target tissue. And by the way, let me ask you again, where are the preganglionic receptors? Where are the preganglionic receptors in an autonomic pathway? Exactly right. Bob said the postganglionic cell body dendrites. Don't tell me that they're on the preganglionic cell. They're not. The receptors are on the postganglionic cell. All right. We didn't finish up the sympathetic. So remember, the preganglionic sympathetic is also cholinergic, but the postganglionic is adrenergic onto alpha and beta receptors on the target tissue. So anytime you see reference to alpha or beta or adrenaline or noradrenaline, you can just cross this one off because we're only talking about sympathetic. Any questions about the general organization? This is just a summary of the overall organization as related to the neurotransmitter systems. You know the craniosacral is parasympathetic, thoracolumbar is sympathetic. You know that the parasympathetic pathway is cholinergic to cholinergic, and the sympathetic pathway is cholinergic to adrenergic. You can see the norepinephrine being released there. This one's a little bit more tedious, but it's basically arguing the same thing. This is the traditional sympathetic pathway here. What are we looking at down here? What's at the bottom? Let me just focus on that one. What is this? Yeah, that's just a regular old motor pathway, so don't be confused. And then here they're showing us that very specialized sympathetic activation of the adrenal medulla. I guess that curvy line is intended to indicate that that's an endocrine release. You won't worry about this one too much. This is the specialized dopaminergic pathway that is also sympathetic. So you don't need to worry about that one. And this one is, all I can say, bizarre. and it will constitute an exception to all of our rules, but for that reason, let's talk about it later. Anybody see anything broad there? The one what's odd about that? Yeah, this is a sympathetic pathway that includes what type of receptor? Muscarinic. Yeah. So we'll mention that again in just a moment when we talk about these receptors in more detail. By contrast, this is your classic parasympathetic pathway here at the top. This is just sort of setting the stage. I think I'll come back to some of these points later. So in this table here, I've listed the adrenergic, these are sympathetic receptors, they're the adrenergic receptors that you need to be familiar with. Alpha 1, alpha 2, beta 1, and beta 2. You don't need to know the second messenger pathway because we haven't had time to talk about that. Later on, you'll be delighted to learn that you need to know those thoroughly, but not now. The only thing to pay attention to now is the location where these receptors are present. And we sometimes argue, although I'm not sure it's terribly helpful, that alpha receptors tend to utilize norepinephrine preferentially. Beta receptors tend to utilize epinephrine preferentially. In my reading, I don't always find that rule holds up. And then you already know, because we're talking about adrenergics, we're talking about amines, this should look very familiar to you. This is the G protein, or second messenger signaling sequence. Now, as I say, the location is important. Alpha 1 everywhere, especially vasomotor. Alpha 2, most important in the GI tract. Beta 1, most important to you for the heart, as in beta blocker. Beta 2 is also all over the place, but notice, well, let's go to the next slide. The function of each of these, again, you don't have to worry about the pathway up here. But what I do want you to know is what it does. And I've circled those points here. Let's circle that one. It's too far at the bottom of the screen. So alpha 1 is a broad spectrum smooth muscle contractor. Alpha 1 contracts smooth muscle, especially in blood vessels. Alpha 2 contracts GI smooth muscle. And we'll talk about this later. Let's analyze the function in just a moment. For right now, I just sort of want you to have a general concept of what's happening. Beta 1, we talked about the heart, so it's going to generally upregulate most aspects of cardiac function. Everything from contraction to conduction and contractility. Probably should add that in there. Strength of contraction is also upregulated by beta 1. And then beta 2 is your universal smooth muscle relaxer. So what do you notice about alpha 1 and beta 2? Yeah, they're antagonistic, and in fact, it's not uncommon to have both receptors in the same tissue. But remember, it's the receptor that determines the function, not the neurotransmitter. All right, let's talk a little bit about the function. And most of this you can reason out. Yeah, I think you have a pretty good idea of how this is going to fall out. Now, in this discussion here, in this sort of table-like thing here, notice I have SNS and PNS, so you know the difference there, sympathetic and parasympathetic. Over here, you have the receptors. So, for example, if we see M, what should you think of immediately? Muscarinic, which is Sympathetic or parasympathetic? Parasympathetic. All right. Rest and digest. So that's the way I want you to approach this. If we look at the heart, for example, and you can see the receptors are beta for the most part, and the way the beta 1 and beta 2 fall out in the heart is much more complicated than we have time to get into it here. But sympathetic activation is going to upregulate or increase cardiac function. By contrast, parasympathetic innervation will tend to depress it. Now, with respect to blood vessel regulation of peripheral blood flow, this is going to be highly tissue specific and receptor specific. So it's on the next slide, but let's consider, for example, sympathetic activation, and you're running. I don't know. You're running from a assailant, and as a consequence, would you anticipate blood flow to increase or decrease in your skeletal muscle? Increase. Now, does that mean that all muscle everywhere, all tissues everywhere, are going to have a comparable increase in blood flow? No. For example, as you're running from the giant anaconda or whatever it is that's coming after you, I heard that we don't have to worry about bears in Florida because some guy was talking about black bears in Florida today. They're kind of scrawny, so you shouldn't be afraid of the black bear. That was his study. They don't get to be more than 250 pounds. So who'd be afraid of a 250-pound black bear? But in any case, if you're being chased by the black bear, what's going to happen to your GI tract? Is it going to upregulate or downregulate? And that would include blood flow as well. That would include secretomotor activity. That would include GI motility. So how is that possible? How can generalized sympathetic activation result in such opposite effects in various tissues? Receptor specificity. So just keep that in mind as we talk about all of these tissues. Beta 2, again, vasodilation, especially skeletal muscle. And alpha 1 are your vasoconstrictors. Alpha 1 are your vasoconstrictors, beta 2 are your vasodilators. The black bear is gaining on you. You continue to dig in and run more rapidly. What's going to happen to the width, the size of your airways? They're going to get larger. Yeah. So sympathetic activation results in bronchodilation. That's a little surprising, perhaps. Make sure you appreciate this. Parasympathetic activation via muscarinic receptors induces bronchoconstriction. Let's not worry about metabolic regulation right now. We'll talk about that later. And we won't worry about renal blood vessels. We'll talk about that later. But the GI tract, we just talked about this. You should be able to work through then the relationship between alpha and beta and muscarinic. Muscarinic, parasympathetic, rest and digest. So the muscarinic, binding of muscarinic receptors is going to enhance GI tract motility and secretion. The only exception to that is that you have in your GI tract, and we talked about this before, an array of sphincters. The sphincters are like valves. So as you begin to smell the warm and dank breath of the black bear at your neck, what's going to happen to those GI tract sphincters? Not the bear's sphincters, your sphincters.
Gonna open up.
What do you mean then? Oh, I was going to say. Probably, although that's the reasons for that aren't thoroughly understood. We can talk about that for a moment if you wish. But no, I just want you to understand that in the GI tract, okay, we've said that sympathetic activation is going to divert blood and basically shut down the GI tract with one exception. Those doorways, those sphincters, are going to clamp shut. So sympathetic activation tends to induce constriction of GI tract sphincters. Everything else is going to take a break. I haven't mentioned paradoxical fear, this bizarre biological response whereby if we get really afraid, we become incontinent. I noticed that before the test in your question. But you know, a lot of animals do that. If you've ever gotten close to one of these lizards out here, if you just look at them the wrong way, they'll drop their tails. Right? If you touch them, they often do that. Birds will shed their feathers when they become very frightened. And so what would be the evolutionary adaptive value of peeing in your pants when you're afraid? Let's say the black bear is on you. Yeah. Does it smell bad? Possibly. I'd want to see the primary research on that. There you go. Well, you get the idea. The bear may get just ticked off. What are you doing? Peeing on me? And it come after you or whatever. We don't know. This is one of many questions. It's paradoxical. It's because it's the reason it gets that term is because it's opposite these normal responses. But in terms of its adaptive value, we don't know. There's a lot of biological literature over the years. I don't know what the adaptive value would be to soiling or peeing your pants when you're under stress. But a lot of animals do it. So with respect to the sweat glands, remember there was an odd one we saw on our table earlier. The thermoregulatory sweat glands, and you've got to be a little careful here because this dichotomy doesn't always hold. But we're talking about sympathetic activation involving postganglionic muscarinic receptors. So this is an exception to our adrenergic rule. There are apocrine stress glands, sweat glands, that do utilize alpha and beta receptors, chiefly alpha receptors. But this is the one that's exceptional.
Is there a difference between M2 and M3?
Yeah, remember in our discussion of muscarinic receptors, we had M1, M2, and M3, M4, and M5. Tissue specificity is the only thing. But what we're really talking about, these are membrane proteins that have slightly different shapes, slightly different conformations, and slightly different responses to stimulation. In terms of those specific differences, you don't need to worry about that. That's far beyond. That's more of a biochemistry. But that's all that refers to. So here we're talking about a muscarinic isoform 3 receptor. All of these receptors have multiple isoforms. We don't need to worry about which. The only ones you need to worry about are the ones that I put in the table back there. The alpha 1, alpha 2, beta 1, and beta 2, because those are so fundamentally different. Now this is called hyperhidrosis. I've known a number of people over the years who feel that this is cosmetically off-putting to their friends and neighbors. They start to, if they experience any stress, they begin sweating profusely at the palms and sometimes at other parts of the body. But this is what is most troubling to some people. How would you address this? You're a PA, you now have thorough knowledge of the autonomic nervous system. Person comes to you for help. What are you going to do? You tell them, yes, I can take care of that overnight. What are you going to do? Okay, you could, but then you might kill them. At the very least, you're going to ruin their day. Well, it would stop a lot of things, though. Yeah, Botox. That's sort of a short term. I want this permanent. What? It may not. So what one, you can go on to Google and find a number of I would encourage you to find somebody doing this in the third world or at an older procedure. Because one way to correct this condition, this is sympathetic. And what do we know about the sympathetic postganglionics? Where do they originate? They're gonna do that in the chain gang for the most part. And you can just go in there and get your pliers, just cut a little hole, get your pliers and crush the ganglion. That'll stop this. Do you see any downsides to that approach?
What if, because ganglia, like, when you have a neurotransmitter, they go up or down, or, I mean, you could inhibit a lot of other.
Exactly, yeah, you're exactly right, and that's often the case. I think they've refined, as I said, they're refining this now, where they can pick specific efferents, but still there is considerable risk. To my knowledge, this procedure still requires collapsing of a lung, for example. So it still requires that a pleural membrane be invaded. They might apply pressure now on how the new procedures go. But I just want you to understand the theory behind the procedure. That's really hyperhidrosis, how to correct it, destroy a ganglion.
Did someone figure out it's T2? How did they figure that out?
Oh, well, like a lot of things, I imagine you try it, over the years, there are different things we've tried, and that seemed to work best. That's how they did my brain surgery. So there's a little discussion here. Yeah, this is good. Yes, I'm sorry.
You said destroy the ganglion, like maybe possibly cut just the white stray line.
Yeah, exactly right, and that's what I was suggesting they actually do now. But even doing that, you're still involving multiple pathways. But you're right, that's exactly what they do. So, but you will also, I've seen the procedures, and anything that you see on the internet, is absolutely true. I've seen where they take the forceps and just turn it. That's that. So we talked about somatic reflexes. Remember, we talked about monosynaptic, and bisynaptic motor reflexes, where we saw the afferent neuron sensing perhaps a candle or pain or something, and there'd be an interneuron that would convey that information to a motor neuron, would be some type of motor response. So that's a somatic reflex. There's just a host of autonomic reflexes regulating every aspect of your physiology right now. Everything from, again, the pupil size to level of perspiration, to whether you want to go to the bathroom right now, and so on. What you should know about these autonomic reflexes, and we'll look at a couple of examples here, is that they're always polysynaptic. They're very complicated. And unlike some somatic reflexes, these require higher processing. They have to go at least up to the brainstem. Heart rate, blood pressure, breathing temperature, all of these would involve polysynaptic autonomic reflexes. Involving, for example, this neuron right here. Somebody tell me, somebody classify that neuron. What kind of neuron is that? Oh, huh? Okay, you're telling me it's a sensory neuron, so that's important. But this is the autonomic nervous system now. So what do we call sensory neurons in the ANS? Yeah, visceral afferents. Yeah, you're looking at one right there. Probably creeping up some piggybacking on some other fiber, and that would be an interneuron, and then you'd have your effector neurons. All right, let's take a break.